While some other folks are joining, I think we're going to go ahead and get started just in the interest of time. So good morning again, everybody, and welcome to our TB 101 series that we've been running this summer, and they'll go every Friday, continue every Friday until September 15th at 10 to 11:15 a.m. My name is Anita Kilal. I'm the Director of Education and Training at the Global Tuberculosis Institute. My colleague Shweta, who's one of our health educators here, is on as well. She's been helping to coordinate this series, as well as Patty Woods, who's one of our TB nurse consultants. And Patty will be monitoring the chat, the questions in the chat, and she'll be reading those off to you, Angela and Atanaska, at the end. To help moderate the Q&A portion. So today you're in for a treat. We know that there are often a lot of questions around lab testing and interpreting of those lab results in the care of TB patients, diagnosis and treatment of TB patients. So actually we added, even though we had a diagnostic presentation last week, we added this session on interpreting lab results because we know that there tends to be a lot of questions. So we're very appreciative that Dr. Angela Starks and Dr. Atanaska Marinova-Petkova are our two presenters today. Dr. Starks is the chief of the Division of TB Elimination Laboratory Branch at CDC. She's been in that position for the past 7 years. In her current capacity, she provides laboratory-based scientific and managerial leadership to the DTBE Laboratory Branch with an interest and research focus in the fields of drug resistance, molecular epidemiology, and molecular diagnostics. She brings broad experience in providing technical assistance for capacity building to laboratories and consultation to TB programs regarding laboratory science and methodologies, including the use of molecular technologies both in the US and internationally. Her branch provides national reference laboratory services for TB, administers a national program for whole genome sequencing for MTB for molecular surveillance, and conducts operations and laboratory research. And Dr. Atanaska Marinova-Petkova is the lead of the National Tuberculosis Reference Laboratory at CDC's Division of TB Elimination. She joined the TB Reference Laboratory team in 2019 as a senior microbiologist and has been in her current role since July of 2021. She serves as a CLIA technical supervisor and oversees the TB drug susceptibility testing service, including the MDDR service, molecular detection of drug resistance, that many of you might be familiar with. The team of laboratory scientists that she currently supervises participate in the drug susceptibility testing of some of the most complicated cases and outbreaks of TB in the US. Dr. Marinova-Petkova holds a Doctor of Veterinary Medicine, Master's in Molecular Biology, and Doctor of Philosophy and Epizootiology and Infectious Diseases and Animal Degrees. Her professional interests include clinical microbiology, molecular epidemiology, drug susceptibility testing, and zoonotic diseases and pathogens. And thank you to both of you for agreeing to present today, and I'll turn it over to you.
Great. Thanks, Anita. Can you hear me okay?
Yes.
Okay, great. Well, thank you so much for the invitation to speak today. Both Atanaska and I look very much forward to the discussions and questions that we will have at the end. But I wanted to start by just providing a little bit of information about our laboratory branch at CDC. We are the Division of TB Elimination Laboratory Branch, and we are dedicated to the elimination of TB, the mission of our division, through a couple of different activities. Those include conducting applied research, providing laboratory services that you'll hear more about today throughout the presentation, and then also directly supporting U.S. public health laboratories to help increase their capacity to combat tuberculosis as well. And we do that through the funding that's provided through a CDC TB cooperative agreement. This cooperative agreement is focused on strengthening public health laboratory services and activities at both the state and local level. And funding is provided to 58 awardees, including 50 state public health labs, the 7 large cities you see listed here, and Puerto Rico as well. So every year when we get our applications in from our awardees from state and local public health laboratories, we collate the data that they provide back to us regarding workload and turnaround time and put those in a tuberculosis laboratory aggregate report that you see reflected here. And that is available on our website. And I just wanted to draw your attention to this first line here in the table where we're looking at workload variables, where we're looking at the number of clinical specimens that are processed. And these are those that are processed just within those 58 awardees that they receive where there is some suspicion of mycobacteria, either non-tuberculous mycobacteria, or in most cases, some individuals that are being evaluated for Mycobacterium tuberculosis. And if you look across from 2017, 28, and 2019, what you will note is that just in these 58 public health laboratory awardees, they are processing upwards of 200,000 specimens each year for that purpose. So a lot of the work that is done in our public health laboratories, as well as the contributions by many of the clinical labs that you all work with and large commercial laboratories as well. We don't have visibility into their workload volumes, but a lot of specimens are going through laboratories and being evaluated. So a few important definitions before we get too far into the presentation. When we talk about a clinical specimen, we are referring to the material that's taken directly from the patient, whether that's a sputum or another potential non-respiratory source like CSF. And these specimens can be raw, which means they're unprocessed, or they may be processed. And we'll talk a little bit more in a moment about what we mean by processing of specimens. When we talk about an isolate, we're talking about the organism that is actually isolated or grown in culture media from a clinical specimen. And then we also use the term from time to time, direct detection. Here we're talking about the detection of nucleic acid, whether that's RNA or DNA, that is specific for Mycobacterium tuberculosis. And that test is targeting the presence of the organism directly in a clinical specimen, hence the term direct detection. This will also sometimes be referred to as nucleic acid amplification testing as well, but it's really that rapid test to detect Mycobacterium tuberculosis directly in a clinical specimen. So some general considerations from the laboratory. Things to keep in mind that not all tests are equal. Tests have different performance metrics, they have different targets, they have different characteristics. And the more test types that are performed within or between laboratories, there is, unfortunately, a higher likelihood that you could have discordance among those results. And I refer back to that first bullet point that not all tests are created equal. It's not that there are bad tests and good tests, it's just that the performance characteristics sometimes differ depending on the particular test that is being employed. Laboratories are also subject to regulatory compliance, and they're also constrained by resources. So there may be times where the laboratory really wants to help, but they're limited in what services they can provide. And so understanding what's available in terms of access through referral is really important because not all the testing, may be available within one single laboratory. And then the final one here on this slide, we all wish that Mycobacterium tuberculosis grew faster. It's a slow grower. Its doubling time is about once every 24 hours. And so I know that you all are acutely aware of the fact that it can take weeks, sometimes months, to get results back for growth-based testing. And so the other thing to keep in mind is that growth-based results, they do take time, especially if repeat testing is needed. So we're going to talk a little bit about expected turnaround times. And it's really, really important that if you are not getting results back within expected turnaround times, that you contact the laboratory and really have that openness for communication so that everybody is aware of what is happening with those samples that have been submitted. So let's walk through quickly the TB testing workflow. This is taken from a new web module that was just recently released. We worked in collaboration with the Association of Public Health Laboratories, and it's entitled Understanding TB Laboratory Testing for Public Health Nurses. And at the end of the presentation today, we'll have a link to that module if you would like additional information. But this is broken out into several different aspects, as you see here, including specimen preparation, detection, that direct detection, either through nucleic acid amplification testing or culture, identification, drug susceptibility testing, both growth-based and molecular, and also supplemental testing that may be performed. For the purposes of today, we're really going to focus on these first four, and then we'll talk a little bit about the MDR service at the very end. So what are those recommended turnaround times? For AFB smear microscopy, it's anticipated that laboratories will turn around a result within 1 day of specimen receipt in the laboratory. For direct detection, that would be within 2 days of specimen receipt in the laboratory. And I just want to point out here, that really these are focused on once the laboratory receives the specimen. In the US, we are still challenged, especially within the public health laboratory system, with quick transport of specimens to the testing facilities if it's going to a public health laboratory. So once they get it in-house, these are the recommended turnaround times. For identification of Mycobacterium tuberculosis in culture, the expected turnaround time is within 21 days of specimen receipt in the lab. However, you need to keep in mind that specimens that have low bacterial loads, where they may be smear negative or a 1+ smear, those are going to take longer to grow than specimens who have higher bacterial loads, a 3+ or a 4+ smear. We did a multi-center evaluation a few years ago where we looked at sort of how long does it really take. And for most diagnostic specimens, if they're going to be culture positive, the vast majority of them will be culture positive within 4 weeks. For follow-up specimens, if they are going to be culture positive, because these would be from patients who are on treatment, so therefore the bacterial load should be lower if they're on effective treatment. If they were going to be culture positive, the vast majority of those were positive within 5 weeks. So just keep that in mind in terms of the length of time that it could take. In terms of first-line drug susceptibility testing, which we refer to as DST, it's anticipated that laboratories would have drug susceptibility test results for first-line drugs within 17 days from the time that an identification of Mycobacterium tuberculosis was made from culture. But again, these are all recommended turnaround times, and it's if all the pieces fit together perfectly. And we know sometimes in biology, those optimal things don't always happen optimally. In terms of mycobacteriology testing in the United States, there are testing services provided through public health laboratories, through commercial labs, reference laboratories, and hospital medical center laboratories. And the work is often piecemeal. We're going to talk a little bit about that, I think, on the next slide, where TB testing may be performed at several different laboratories to accomplish a full panel of testing. Different laboratories, for example, perhaps hospital medical center laboratories, may only perform AFB smear, and it has to be referred somewhere else for culture, perhaps somewhere else for identification, and even somewhere else perhaps for drug susceptibility testing. But again, communication is really key, especially when the testing becomes further removed from the originating laboratory. I talked a little bit earlier about the need to have awareness of where to obtain testing. I think that's particularly important, especially as sometimes we do have a need to refer to multiple laboratories. This is especially true in more complex cases where there may have to be, just out of necessity, testing at more than one laboratory, especially when we see drug resistance. There are, as I mentioned earlier, differences in methods and test performance. And there are differences in how results may be reported. And I'm sure that you all are aware of that in some of the reports that you have seen. Those differences can impact sometime turnaround times for results to healthcare providers, whether reporting happens electronically or manually, or sometimes perhaps even through snail mail. Those things can all impact turnaround time. And differences in how laboratory reports are formatted, or the terminology that's used, or even the nomenclature, can also serve as a potential source of confusion. We do not have a standardized way in which we all are reporting laboratory results. So these are all just things to keep in mind. So let's talk about specimen collection. Sputum is the most common specimen type when an individual is being evaluated for pulmonary tuberculosis. And for diagnosis, it's recommended that 3 specimens are collected 8 to 24 hours apart, with at least one of these being an early morning specimen. For follow-up specimens during treatment, it's recommended that specimens are collected at least monthly for smear culture and repeat drug susceptibility testing if needed, if the culture remains positive after completion of the intensive phase of treatment. The patient should be instructed about proper collection, and specimens should be collected in sterile, leak-proof, disposable, non-breakable, appropriately labeled, lab-approved containers, and the laboratory that you're submitting to should be able to provide you with the information regarding what all of the requirements are. It's also really important that specimens are rapidly transported to the laboratory. They should be shipped to the laboratory and received at the laboratory within 24 hours of collection. But in the event that that doesn't happen, it's really important that the specimens are maintained in proper storage conditions at refrigerated temperatures to avoid overgrowth of other bacteria that could be contaminating the sample. And you see here on the picture an example of a poor quality, which would be very watery or salivary in nature, versus a good quality specimen that is going to be that really thicker mucoid material. And that's really what we're after in terms of a good quality specimen, sputum specimen in the laboratory. So really it's in terms of sputum specimens, or any specimen for that matter, it's garbage in, garbage out. Really, if we don't start with a very good quality specimen of sufficient quantity, it's going to be really hard for these laboratory tests to do their optimal performance. And so we don't want to process poor or suboptimal specimens, because it is a burden in terms of both financial and personnel resources. Again, results generated from processing suboptimal specimens may not be reliable. And as I mentioned, each laboratory will also have its own rejection criteria, and these should be readily accessible to submitters. So those are things to keep in mind with the laboratories that you're working with. And clinicians should be notified when a specimen is rejected, and the reason for that rejection should be provided. Just generally, specimens collected by invasive procedures should not generally be rejected. So possible rejection criteria that you might encounter. Really, it's that we call it the pre-analytical phase of testing. This is where the specimen is collected and handled and transported. Everything has to be properly labeled. There's a process that we work in the laboratory where we're going to try to match all the paperwork with what identifiers are included on the specimen, and we have to make sure those match, or we can't proceed with testing. We also, in the laboratory, often have minimal volume requirements. That may be a reason for rejection. Swabs in general are not something that are generally accepted for mycobacteriology testing for the reasons that you see listed there. It's generally not advised to pull sputum specimens or other specimen types if the sputum has been left at room temperature for extended periods of time. And then if there's a broken or leaking container, that is certainly a place where the laboratory would reject that sample. Excessive delay between specimen collection and receipt in the laboratory. I know some of our public health laboratories have limitations where if the specimen is more than 5 days or 7 days from collection, they won't accept it. Blood specimens, you see listed here, fixed tissue, and then gastric lavage if the pH has not been adjusted to neutralize that within 1 hour of collection. These are all reasons why a sample could be rejected. In terms of specimen types, you see listed here on the left-hand side respiratory specimen types that would be commonly submitted for TB testing. And then on the right-hand side, some non-respiratory specimen types that might be submitted for evaluation. So what do we mean by processing? And I'm going to talk specifically about pulmonary specimens. We often talk about digestion and decontamination. And this is done because when we have a good quality sample that is very thick and mucoid, we really have to break up that material to get optimal recovery of the mycobacteria in that sample. And to be able to do that, we have to do what's called digestion that you see listed here on the bottom left-hand side. So we use a mucolytic agent to release, the acid-fast bacilli that may be present in that sample and expose the normal flora to decontamination. And often that includes the use of sodium hydroxide. And so the decontamination step is used to kill the normal flora that might overgrow the slow-growing Mycobacterium tuberculosis. But one thing that's really important is that the decontamination step not only can result in death of normal flora in the sample, but also 30% or more of the mycobacteria can be killed during processing as well. So it's really important to have the timing of that decontamination step to be very strict so that we don't result in lowering whatever the numbers of mycobacteria that may be present more than necessary. And then there's a centrifugation step that's used really to concentrate everything down. This process, there'll be a buffer to use to neutralize everything spun down, and then it'll be resuspended, and that's when the workflow would continue. Usually with the next step, which we're going to talk about, which is acid-fast bacilli smear microscopy, or AFB smear microscopy. So AFB smear microscopy is a rapid and inexpensive method that's performed to detect AFB. And globally, it's still used, although I will say there has been a global shift even in use of more molecular technologies. But because it is rapid and relatively inexpensive, it is still often used as a first step in the mycobacteriology workflow. It uses specifically stains that specifically target acid-fast organisms. They're examined under a microscope, and you see some examples here on the top, on the right-hand side of fluorescence microscopy, and then on the bottom of the Kenyan staining. But keep in mind that both non-tuberculous mycobacteria and Mycobacterium tuberculosis complex are going to be acid-fast. So acid-fast bacilli smear microscopy has relatively low sensitivity, and it is not specific for TB. In fact, in some of our Midwestern states, and perhaps even in some of the states that you all work in as well, we would see just as many non-tuberculous mycobacteria as we would see Mycobacterium tuberculosis, in some cases probably more. And the reliability of this test really depends on the number of AFB that are present in the specimen. In terms of reporting language for AFB smears, this is just a couple of examples of different grading systems that are used. Some laboratories may say no AFB seen or report as negative. Others may say 1 to 2 bacilli seen or equivocal or order repeat specimen. And then as the positive results are reported out, they could be reported out as 1+ to 4+, dependent on the number of organisms that are observed in the field under the microscope, or anywhere from rare, few, numerous to many. So it really just kind of depends on the grading system that the particular laboratory is using. In terms of the sensitivity of microscopy versus culture, for a positive smear, you need somewhere between 5,000 to 10,000 AFB per mil. Culture is more sensitive, where you only need 10 to 100 to have a positive culture. And in the United States, only about half of our cases are originally smear positive. So that really lends itself to, talking about the significance of culture here. Culture is still the gold standard. It really is important for confirming tuberculosis, and having an isolate is really important for drug susceptibility testing, genotyping that may be done to understand clusters or potentially outbreaks to identify those, and also to evaluate the response to therapy. In the US, about 80% of our cases of pulmonary TB are culture positive. Others are culture negative and are either a clinical or provider-provided diagnosis. And you see some examples here on the right-hand side at the top of a BD MJIT tube. that we use for liquid culture. And on the bottom, this looks like it's probably, I'll call it a 7H10 Middlebrook plate that we would use for inoculating and observing growth on solid media. So now we're going to move to more of the molecular testing. I talked earlier about what is meant by direct detection. Direct detection is really focused on rapidly detecting Mycobacterium tuberculosis complex directly in a clinical specimen. And we do often refer to this as nucleic acid amplification testing as well. As I mentioned earlier, the turnaround time should really be within 2 days after specimen receipt in the laboratory. And if our turnaround time is longer than that, it really starts to negate the importance of the rapid nature of these kind of tests. Positive results demonstrate the presence of Mycobacterium tuberculosis complex in the sample, but a positive result does not distinguish between live and dead organisms. You could have an individual who could be shedding dead organisms for quite a while, and these nucleic acid amplification tests would actually pick that up as a positive result, but it does not give you any information about the viability of the organism. Likewise, a negative result does not necessarily mean that the absence of Mycobacterium tuberculosis complex. There can be things in the sample that can inhibit the PCR amplification that's needed for these tests. Additionally, the amount of material that's available could be below the limit of detection for the test. And this is important because, it's more likely to detect Mycobacterium tuberculosis when the number of organisms in the original sample is higher. It's not useful generally for monitoring treatment response for some of the reasons that we just discussed. And clinical judgment is still needed to guide decision-making to treat individuals based on these results, or also in use of these results for isolation practices. But the sensitivity is quite good. For AFB Smear positive TB patients, for most of the tests, whether they're the commercially available Gene Expert NTB Ref assay or laboratory developed tests like real-time PCR, in AFB Smear positive samples, the sensitivity tends to be 95% or greater. There is some decrease in the sensitivity in smear negative samples, and again, that correlates to there being fewer organisms and less target available in those types of samples. But the sensitivity still is pretty good, somewhere between 55 to 75%. And if you test more than one sample in a smear negative patient, sometimes you'll get an incremental increase in sensitivity. So a few other considerations for culture versus these rapid nucleic acid amplification tests. The rapid detection is really key for patient care and public health. It helps us to quickly identify these patients, to help to get them on effective treatment, and also to be able to put in place any public health interventions that may be needed to prevent ongoing or potential transmission. It's not yet able to replace culture, the molecular test that we have available. Culture still remains our most sensitive method, and we talked about why having an isolate is needed. Some TB patients will have a negative culture and a negative nucleic acid amplification test. That can happen, but they can still have tuberculosis. And that goes back to the statement I made at the very beginning that, laboratory tests are not perfect. The laboratory also may not have validated the use of these types of molecular tests for other respiratory sample types other than sputum, and also may not have validated the use of these tests for extrapulmonary sources. For the Gene Expert NTB Ref assay, it is specifically approved in its instructions for use for the use of testing of sputum samples only. Currently, a laboratory can do off-label use of an FDA-approved assay, or they could validate different matrices for use with that test, but they would have to, in their own laboratory, conduct a robust validation and determine those performance characteristics before they could do that. And in the US, it's oftentimes quite difficult for laboratories to be able to obtain enough different types of material to conduct those types of validations. So that's why in some instances, laboratories may not test for all the different specimen types that they would like to. Testing for pathology samples when the sample will not be viable for culture may be an option. And we have here at CDC, the infectious diseases pathology branch that we work very closely with that has the ability under some circumstances, and they have very strict criteria, but to accept formalin-fixed tissue, they can do DNA extractions and look at pathology to determine if tuberculosis is a diagnosis. And then they can do molecular testing to identify Mycobacterium tuberculosis in those samples. And in some instances, we have been able to work with them to be able to get some molecular testing to provide something regarding susceptibility as well. So as I mentioned, there's currently only one FDA-approved test for use with sputum only, and that's the CEFI Expert NTB Ref. And in its instructions for use, it indicates that this test should only be requested if a patient has been on treatment less than 3 days. So it would be very early when the individual is being evaluated for TB. There are other non-FDA approved tests that some laboratories use, including things called line probe assays. And as I mentioned, there's also laboratory developed tests, or LDTs, which are used by a number of laboratories as well. And these may include things like real-time PCR or DNA sequencing assays. So what patients or specimens should be tested by these molecular tests? Well, our guidance says that really, if you think the patient has tuberculosis, if they have signs and symptoms consistent, that a specimen from that patient should be tested by nucleic acid amplification testing. The laboratory will not always know this information. And so in some cases, they have algorithms in place where they may only test new smear positives. And that's because of that increased sensitivity and concerns about infectiousness. And they may only test smear negatives on request. So if you have need to have nucleic acid amplification testing performed, that communication with the laboratory is going to be critically important. Unfortunately, we still are lacking in terms of use of this technology for some of our TB cases. This is a study that we conducted a few years ago, and I'll just draw your attention to 2017. And the yellow is those TB patients who had a nucleic acid amplification test performed that had a negative result. In the red, those that had it performed that had a positive result. And then unfortunately in the blue here are those where there was no report of having had this testing performed. And that was over 30% of our cases in 2017. And I don't think that's improved very much in recent years. So there's potentially a missed opportunity there for rapidly detecting TB cases. In 2015, the US FDA approved expanded claims for Expert NTB Ref related to its use for guidance in airborne infection isolation decision-making. And APHL, the Association of Public Health Laboratories, worked with the National Tuberculosis Controllers Association to develop a consensus statement, and they issued this guidance in 2016. And basically what this is focused on is that a negative result using nucleic acid amplification testing from 1 or 2 sputum specimens was as predictive of results of 2 or 3 AFB smears being negative. But again, there was a statement in here that sputum test results alone should not be the only criteria for clinical decision-making in regards to AII. So let's move to culture. I've mentioned the different culture methods that we have. Typically, the processed specimens will be inoculated to both liquid and solid media. The ones that may be most common in terms of solid media that you've heard of are Middlebrook agar or LJ, Lowenstein-Jensen slants. And the advantage of growth on solid media is the colonies can be visible on the surface of the media, which is helpful for seeing if the phenotype is consistent with Mycobacterium tuberculosis. Most laboratories will hold solid media somewhere between 6 to 8 weeks before calling that negative if they don't see growth, but it can take longer for the organisms to grow on solid media, which is why we use liquid broth media systems. And the most common that you'll probably encounter is the MJIT that's listed first here. As I mentioned, we have faster recovery in liquid media than on solid, and the incubation default for these liquid broth automated systems is 6 weeks. So you would not expect to receive a final negative report for growth in liquid culture until they have incubated for at least 42 days. And they may not even issue the laboratory a final negative report until the solid media has been held up to 8 weeks. The current recommendation is that at least one piece of solid media is inoculated in combination with the liquid culture. So MTBC, as I mentioned, is slow growing. We talked about the fact that most diagnostic specimens, if they're going to be positive, will grow within 4 to 5 weeks, and that follow-up specimens can take a little longer. Specimens that have a higher number of organisms will turn positive faster than those that have a lower bacterial burden. And if the culture is positive, as indicated by the automated systems that we have for liquid culture, the next thing that will happen is that there will be typically a sample will be taken to examine under the microscope to determine if those are acid-fast organisms that are growing in that culture versus some sort of contaminant. And then if that is consistent with Mycobacterium tuberculosis, a sample would be taken to make the identification of what's growing there. We call that ID for short. And then from there, if it's Mycobacterium tuberculosis, then we would move on to drug susceptibility testing. So these are common methods that are used for identification or ID in the mycobacteriology laboratory. They include MALDI-TOF or mass spectrometry, DNA sequencing of specific targets for Mycobacterium tuberculosis, real-time PCR assays. I mentioned LPA earlier, and our laboratories may also use the CEFI Expert NTB Ref, although it's normally used for nucleic acid amplification testing very early to direct detect in the clinical specimen. Some laboratories have validated its use to be able to do identification from culture as well. Additional methods you may encounter include high-performance liquid chromatography, or HPLC, although there are fewer laboratories using that these days. And then, unfortunately, the most common method, which was pretty straightforward, the hologic AccuProbe, was discontinued in late 2022, and laboratories are currently working through their final stocks of that particular method. So you're more likely to encounter some of these other ones that I mentioned above. It's also important to keep in mind that there can be false positive results. It does happen in the laboratory from time to time. And we developed a few years ago with APHL an interactive web module where you can learn more about false positives and how to work through a potential false positive investigation. And so this will be available to you as a resource after you receive the slides. So let's move to drug susceptibility testing. I'm going to start with growth-based drug susceptibility testing. Some drug susceptibility test methods test at what we call a critical concentration. And this is the lowest concentration of an anti-tuberculosis drug that will inhibit growth of most strains of Mycobacterium tuberculosis complex that are susceptible to that drug. This would be a single concentration that could be used to separate susceptible from resistant organisms where treatment with that drug would be unlikely to be successful. And so typically when a laboratory is testing rifampin, isoniazid, ethambutol, PZA, your first-line drugs, they're going to be testing it at a single critical concentration and providing you back with a categorical result of resistant or susceptible. There are, however, laboratories that are increasingly evaluating and potentially even using another growth-based DST method that some of you may have familiarity with for other bacteria. Includes the use of minimum inhibitory concentration, or MIC testing. In this testing, the MIC value is the lowest concentration of an antimicrobial drug that prevents growth, visible growth of the organism. And so in these cases, we do not have breakpoints, formalized consensus breakpoints for minimum inhibitory concentration testing. So it can be difficult sometimes for laboratories to provide, if they're doing this testing, a clear categorical result of resistant or susceptible. And in place of that, they may instead just report an MIC value in terms of the concentration of drug that inhibited growth of the organism. They may provide that itself in the laboratory report. So what you will see really kind of depends on what the testing that the laboratory is doing, what test method they're using. But in most cases, laboratories are doing critical concentration testing where they can provide a categorical result. Okay, so it's currently recommended that all isolates of Mycobacterium tuberculosis, so if you have a culture positive patient, you have an isolate, that the initial isolate be tested against all 4 primary or first line drugs. That includes rifampin, isoniazid, ethambutol, and PZA. The mantra is changing a little bit because of the increasing use of fluoroquinolones in terms of right now, we've had some experience with drug shortages for isoniazid where fluoroquinolone testing has become incredibly important because of the need to add a fluoroquinolone. Additionally, with the recently reported 4-month drug regimen that can be used for drug susceptible TB, moxifloxacin, a fluoroquinolone, is an important part of that panel as well. So fluoroquinolone testing is becoming increasingly more important and may actually at some point be considered as part of the first line panel that would need to be, for which isolates would need to be tested against. If an isolate is found to be rifampin resistant, indicating, in that case, a need for an MDR regimen, or resistant to any 2 first-line drugs, it is recommended that those isolates are reflexed for second-line drug testing. Those can include fluoroquinolones, either levofloxacin or moxifloxacin. Second-line injectables, those are being less frequently used for treatment, and with the changing definitions that we've had for extensively drug-resistant TB, perhaps fewer laboratories are testing those, but we still test them here at CDC. And increasingly, there's a need for access to testing of newer and repurposed drugs, including bedaquiline, clofazimine, linezolid, and protomidate. So all of these are very important needs in terms of being able to perform drug susceptibility testing. As CDC and in a few of the laboratories that still do second-line testing, we have about 20 in the US that still do second-line testing, agar proportion is one of the methods that's often used. We use a quad plate, and as you see here, we would have a no drug control. That's on the far left side in the upper corner, this particular quadrant that has a lot of those little asterisks in it. That's the no drug control. We should see growth there. And then you would have 3 other drugs on this plate in this example: isoniazid, rifampin, and streptomycin. In this particular example, you see that we have growth on the INH quad and also on the rifampin quad, indicating that in this particular example, this is likely to be a multi-drug, at least a multi-drug resistant isolate with those 2 resistances. Some laboratories, as you'll see here in the bottom, may use more of like a disc diffusion type of testing. And then on the right-hand side is an example of our picture of the BD MJIT instrument that we use for that automated liquid culture system that is also used for primarily for first-line drug susceptibility testing in laboratories doing that. Here's an example of growth in a microtiter plate that would be part of that MIC testing that I mentioned earlier for determining minimum inhibitory concentrations. And you see the drugs listed across the bottom, and then increasing drug concentration from the bottom to the top following that arrow. So basically in this first column for ofloxacin, the lowest concentration of drug would be that second circle from the top. That would be the MIC value. It's the lowest concentration of drug that inhibits visible growth of the organism. And I would say that increasingly laboratories are evaluating more use of MIC testing as clinicians are asking to have these values, especially in very complex drug-resistant cases. So now let's shift a little bit to talk about molecular detection of drug resistance, or molecular DST. This is focused on examining the DNA of specific genes for mutations that are known to be associated with phenotypic resistance. And really, we have to ask ourselves, where are the mutations in what genes that we know are associated with resistance? Where are the mutations within that gene? And are there hotspots? We know, for example, with rifampin resistance, there's the RPOB gene. And within the RPOB gene, there's a region called the RRDR, or rifampin resistance determining region. And most of the rifampin resistance that we're going to see is associated with mutations in that hotspot. Newer methods are becoming the standard methodology. I will draw your attention to Wadsworth Center in New York State. They are now using, and have been for a number of years, whole genome sequencing as their primary method. And that helps with turnaround time. It helps with more comprehensive information. And really, they've been able to replace about 80% of their growth-based testing in favor of whole genome sequencing. And we do know that patients are being treated increasingly based on molecular DST results. On this right-hand side, the World Health Organization came out a few years ago with a catalog of mutations in Mycobacterium tuberculosis to help provide additional information about mutations and their association with drug resistance. This was version 1. We've been contributing, as have others, to the completion of version 2, which will have even more information, and I think will continue to drive the use of molecular testing for this purpose. So what are some considerations for molecular drug susceptibility testing, or also known as molecular detection of drug resistance? Again, as we talked about for other assays, the assays for molecular detection of drug resistance are not necessarily equal. There are different performance characteristics. There are differences in the loci that may be examined, or the sample type that could be tested, in the output and the results. It's important to understand the information that is being provided by the test, what its limitations are, and also the expected turnaround time. And I reiterate again the critical importance of communication between the laboratory and the healthcare provider. It's also important to keep in mind that there can be what we call hetero-resistant populations. And so this would be within the same sample, a mix of susceptible and resistant organisms, as sometimes these hetero-resistant populations, especially if there are very low numbers or low proportion of resistant organisms, it could result in discordant results between different molecular methods, and also discordant results between molecular and growth-based drug susceptibility testing methods as well. Whole genome sequencing, while very exciting and very powerful, it doesn't help to solve everything, because we still have to know what we're looking at with the output, regardless of the test that's being used. And what is true for one drug may not be true for another. I remember when I gave these presentations 10 years ago, our mantra also always used to be that if it's a silent mutation, it does not have any effect on resistance. Well, we've had to stop saying that, because a few years ago, we found out that sometimes a silent mutation, and I draw your attention to the last line here, it's a leucine 203 leucine. It just tells us where the change was. There's a change in the DNA sequence, but it does not result in a change in the amino acid sequence in the resulting protein, hence the leucine 203 leucine. It's the same, right? That particular mutation, which surprisingly we found, actually does result in isoniazid resistance. And it's part of our testing at CDC, as in other laboratories. So we can no longer say silent mutations don't result in resistance, but generally they don't. We just have recently found this example where at least one, it does. Okay, so what are the potential effects of these mutations? Well, they could have no effect. They could have a change in the protein structure that inhibits the drug activity. And that may be something like CATG for isoniazid, where you end up not having activation of the drug. It could result in a change in the protein structure so that the drug cannot bind to its target. It could have a change in a regulatory region that changes the expression levels to overcome the effects of the drug. It could result in a change in affinity for the drug. And it could even, especially, the example that comes to mind is in rifampin. Different amino acid substitutions can result in different levels of resistance to rifampin. So all of these things can have impacts. And there are different molecular drug susceptibility testing methods. The CEFI Gene Expert NTB Ref assay only focuses on rifampin, and it is not a sequencing-based assay, it's a probe-based assay. So the information that you're going to get from something like that, and also from the line probe assays, is going to be quite different than the results and output you're going to get from a DNA sequencing assay. And it's currently recommended that if any of these first two tests, either the NTB Ref assay or a line probe assay, give an indication of resistance, that a sample is referred to another laboratory that is performing DNA sequencing to be able to understand more about that mutation, to confirm the presence of the mutation, and to understand a little more information about what that mutation may be resulting in terms of resistance. So with that, I'm going to turn it over to my colleague, Dr. Adinoska Marinova-Petkova.
Thank you, Angela. So as we already mentioned, the molecular methods are looking into the mycobacterial genes that are known to confer resistance for particular drugs. In this table, we're showing on the left side the drug name, and on the right side of the table, known genes for resistance that confer resistance to the particular drug. So if you look at the first line drugs, you will notice that rifampin, pyrazinamide, and ethambutol have one known gene that we're looking into for resistance, versus isoniazid, for example, is a little bit more complex. We know at least about 4 different genes that can confer resistance. In our lab, we're looking at the first 3. Out of all of these first-line drugs, rifampin is probably the most straightforward, because we know that there are 3 main hotspots within the RPOB gene that if we look into and we don't find mutations, we can almost with 100% certainty claim that this is going to be phenotypically susceptible, versus with all the rest of the drugs, as well as with the other second-line drugs and the new and repurposed drugs that I will show you in a second, even though we may not find mutations within those regions, there are other mechanisms of resistance, known and unknown, and other regions and genes that may be contributing to resistance. So just because we didn't find mutations doesn't mean that we can fully rule out resistance to those drugs. We have listed here some of the second-line drugs, ethionamide, rifabutin, streptomycin. So, for example, RPOB is responsible for resistance to rifampin and rifabutin as well. And there are mutations that are known to confer resistance to rifampin, but can still allow the provider to use rifabutin in the patient treatment. They don't necessarily confer resistance to both. How do I switch to the next to the right? Oh, okay, thank you. Here are listed the rest of the second line drugs that we look into in our lab, fluoroquinolones and second line injectable drugs, as well as the new and repurposed drugs. So I am glad to say that since February of this year, as I will show you in a minute, we're able to look into the genes for bedaquiline, linezolid, and clofazimine and report mutations that are found there. In terms of predominant and delaminate, these are not part of our panel as of now, but we're working to incorporate them in the future. Okay. A few words about the CEFI Gene Expert NTB Ref assay. This is, as you probably all know, automated commercial system for identification of M. tuberculosis complex and mutations in the RPOB gene for rifampin resistance. This is an FDA-approved test, but I should say that it's FDA-approved only for sputum samples, and if a lab would like to test other specimens, that is now a lab-developed test that requires additional validation for those sample types. It's in its essence, this is a real-time PCR assay that uses molecular beacons. These are 5 probes that overlap with each other to cover the RRDR hotspot within the RPOB gene. And with that being said, I should really mention here that this is not the only region. There are 2 more hotspots for rifampin resistance within the gene that GeneXpert does not look into. It works with decontaminated and digested samples, but it can also work with raw sputa. It does the DNA extraction, amplification, and all the rest of the steps within this single cartridge that is shown on the left side. It takes about 2 hours time to complete the run, and it's a minimal hands-on manipulation, which is technically very simple for processing. Let's talk a little bit about the CDC Molecular Detection of Drug Resistance, or MDR Service, that probably many of you are familiar with. This is a service that was implemented in our lab and branch back in 2009. And it is a clinical testing service that provides rapid detection of drug resistance to TB, as well as confirmation of MDR cases that were detected in the labs throughout the country. It provides additional information for second-line drugs as well, because many of the labs in the US do not perform second-line drug testing. It is available to all 50 US states and territories, as well as the affiliated Pacific Islands. It's free of charge, and the shipping costs are covered through APHL account with FedEx. We also offer clinical consultation on request, which is specifically on the interpretation of test results that our lab have provided. In February of 2023, a few months ago, we transitioned the MDR service from pyrosequencing and Sanger sequencing to a new targeted next generation sequencing assay. And if you want to know a little bit more about that, the link at the bottom of the page can take you to the MDR user guide on our website that has a lot of information about it. The acceptable sample types, just like previously, are confirmed MTBC isolates, pure cultures, but also mixed and non-viable MTBC cultures. We do accept nucleic acid amplification test positive processed sediments as well. And this is the reason why we didn't go, one of the reasons why we didn't go towards whole genome sequencing, but stayed with targeted next-gen sequencing, because it allows us actually to continue to test processed sediments. We don't accept DNA that was extracted by external to CDC partners, but we have long-standing collaboration with the CDC Infectious Diseases Pathology branch. So in certain cases, when the providers have formalin-fixed paraffin-embedded tissue blocks from a patient with high suspicion for TB, they can contact directly IDPB at CDC. And if they accept, they have their own set of acceptance criteria, but if they accept this sample and if they confirm TB in it, they can forward DNA extract to our lab, and we provide pyrosequencing for rifampin and INH on those specific DNA extracts. More information about collection, storage, and transport, again, you can find in our MDR user guide on our website. This is the testing panel of what it looks like currently with the TNGS. It has all the first-line drugs. From the second-line drugs, we have fluoroquinolones, second-line injectables. And from the new and repurposed drugs, we have now included bedaquiline, clofazimine, and linezolid, which we haven't been able to test previously. One additional thing is that for isoniazid, the CAT-G gene, previously we were only able to look into the region of the gene that was including the 315 codon mutation. And now we are looking at much larger regions. So we've already started seeing additional mutations that we weren't able to detect previously. Right. Okay. This is the MDDR algorithm. It has not changed since the introduction of TNGS, but just briefly, we receive the specimens or the isolates at CDC. And I should just mention that we don't directly receive your samples. They go to, we have a centralized accessioning at the STAT laboratory. And then after that, our lab receives the samples for testing. We look first into all the paperwork, acceptability. We document all of that. If there is anything that needs to be corrected, we contact the submitters again. And until the paperwork is cleared, we cannot test the sample, basically. So after the paperwork has been cleared, we initiate molecular testing and growth-based testing in parallel. For the molecular testing, we do TNGS now for all the samples, except for the DNA extracts that I mentioned that go for pyrosequencing and certain loci by Sanger sequencing. And the turnaround time for the preliminary report for the molecular result is currently 7 to 10 days, which is a little bit longer than the previous approximately 4 days. However, there is a good reason for this. First of all, this turnaround time is inclusive of any holidays, weekends, as well as the time that the sample has spent at the STAT laboratory. The clock starts ticking from the time sample is received at CDC. As well as the run itself includes 10 samples. So before we have 10 patient samples, we cannot initiate that. And it's a little bit more hands-on and labor intense. It takes approximately 3 days just for the wet lab to be completed, along with the results to be analyzed. We now report the molecular results through the ELIMS system, which is a new thing. And this is when you receive the molecular results preliminary report. The growth-based DST, if everything goes well, it takes approximately 35 days, but please know that for specimens that take longer to grow or samples that don't like to grow on this media and need repetition of the test may take a few additional weeks until we can issue the final report. This is how the previous format looked like on the left versus the new ELIMS format for the molecular testing. We got a lot of positive feedback from submitters about how this was simplified and how this flows along with the phenotypic results that we previously reported through ELIMS. So basically, anything that is a open reading frame mutation is now only reported as amino acid change versus nucleotide and amino acid. And we have simplified the interpretation of the results significantly. I'm not going to go through all of this. We have listed what you see on the reports, and I'm hopeful between February and now if you have submitted something to us, you've already seen one of our reports. But basically, you have all the patient information on the front page. Then for the molecular preliminary report, you will see all the drugs for the phenotypic drug susceptibility testing listed, and they will show as pending and the time of the molecular report. And then you will see what I just showed for the molecular, along with some disclaimers and comments at the very end of the report. I wanted to point out that we work directly primarily with the public health laboratories, and we're only authorized to directly send the reports back to them. This is how the ELIMS system works, and this is how CDC works. So this impacts for sure the turnaround time between the time the sample was collected and the time you receive the final report. So basically, if a provider from a private practice or a hospital lab needs to submit something to us, it needs to go through the public health laboratories, and this is who they will likely receive the report from. If you need to submit a sample to us, on the right side here, I'm showing what the molecular detection of drug resistance request form looks like. And we always look to see the public health laboratory contact information in the top page, because this is who we contact for the results as well. This is available through our website. This is where you can find this. And this form is submitted to us by email or through the CSTOR portal for pre-approval. We look at everything and then we send additional information and acceptance criteria and some guidance for shipping and labeling the samples back to whoever is going to send the sample to us. This is the 5034 form, which you're probably very familiar with. We see this form with the packaging that we receive from the public health laboratories, and we also request that the MDDR request form is attached to this, so when we receive it, we can match the information to what we've pre-approved. We recently introduced at the end of May the rapid testing for fluoroquinolone resistance, and this was prompted by specifically the INH shortage that is nationwide now, as well as the 4-month rifampicin moxifloxacin regimen. So what is the rapid fluoroquinolone testing? This is a condensed Sanger sequencing panel for rifampin, INH, fluoroquinolones, and PZA, only molecular testing. But if we find mutations that are reportable for rifampin and or fluoroquinolones, we directly internally reflex this to full panel MDDR testing, including TNGS, as well as phenotypic drug susceptibility testing, and we follow up with that report. And I'm going to turn over to Angela just quickly to talk about the DST Reference Center.
Yeah, I just wanted to draw your attention to another resource that we have available, which is the DST Reference Center at the California Microbial Diseases Laboratory. This was established to support laboratories, public health laboratories that had low volumes of drug susceptibility testing. And if you'd like additional information, the URL is listed there. Those services performed by this laboratory are complementary to what we do in our laboratory, and they do not replace the services that we perform here. They're just done in addition to. So these, not knowing what states you all are from, but this gives you an idea of who's submitting what to us. Some of the laboratories, primarily in the middle part of the country and up here in the Northeast, have lower volumes of testing, and they do send things primarily to California to get both growth-based and molecular drug susceptibility testing. And then we have a few other locations that may just use the services in California more ad hoc basis, depending on what the needs are for the laboratory. But this is something that is available to states that have a need. And then lastly, I just wanted to mention the web module that I talked about at the beginning, and you saw some of the slides from Understanding Tuberculosis Laboratory Testing for Public Health Nurses. The URL is listed there, so if you would like additional information, this would certainly be a resource that's available to you to sort of dig in a little bit more. And with that, here is the contact information for both Adenoska and myself. We're very open to answering questions and to helping to connect resources and doing anything that we can to be helpful. So always feel free to reach out to us. We want to be helpful in any way that we can. And we really appreciate the invitation today. Thank you so much.
Thank you. Sorry, I've got my camera. Okay. It's not working. Okay, there it is. Thank you so much for that presentation. It's always, I have a lot of questions, but this was a really good segue into when you were talking about the resource for public health nurses, and the TB Lab link is in there. I need to just put it in there, but we are having a webinar. The National TB Controllers, it's not controllers anymore, Association and Nurse Coalition is going to have a webinar about that resource and how to use it, and that's going to be August 18th. I believe it's 12 o'clock, but anyway, look for that information, and it'll be coming out shortly to register. I do have a question though. In one of the slides where you had the detection and you had Gene Expert, PCR, and there's a bunch of other tests. Are all those tests considered NAT tests? Like, is a PCR also a NAT? Like, I know a Gene Expert's a NAT, but like when we get, that's something that we do have to report when, to CDC, and I think some of the nurses I know in my state get a little confused of when they see different reports from hospitals, they're not considering it a NAT because it doesn't say NAT, but it's.
Yeah. This goes back to that we don't all report things in the same way, and it can be confusing. So PCR is just a method, right? It's a method that could be used for nucleic acid amplification testing for direct detection from a specimen, or it could be used for identification from a culture. So really, when you're looking at the report, you have to understand a couple of clues when you're looking at the report. Would be the time frame from the specimen collection to the report of that result. If that's within a short time frame, let's say the 3 to 4 to 5 days from specimen collection, that's a pretty good indicator that that is a result from a nucleic acid amplification test, right? Whether it's Gene Expert, whether it's a real-time PCR assay, whatever that methodology that was used. The other thing to look for is that if the time frame is longer, say from specimen collection to the result is weeks, that is not likely to be a nucleic acid amplification test. That's likely to be an identification from culture. But one way that you can look at that is if you look up, usually reports are cumulative, at least most of the ones that I've seen. So you could kind of look either up or down, depending on how they're sorted in the report, to see if there was a result of AFB positive growth or something about culture, to understand exactly what you may be looking at, whether it's nucleic acid amplification test or identification from culture. Because we can, unfortunately, use the same kind of methods for both.
Right.
So those are the kind of the tips and tricks to kind of understand what you may be looking at in terms of the result.
I like that. That is a great suggestion. That's going to be very helpful, I'm sure. The other thing is sometimes you'll see like DNA probe. And I know like you had DNA detection, but if you see a DNA probe, is that different than a NAT test or?
Good question. I hate that we use that terminology, DNA probe, because again, it causes a lot of confusion. And unfortunately, we can talk about it for both. We can talk about it. It's kind of the same rules apply, which we just talked about, really looking and digging into the laboratory report and sort of understanding those timeframes. But generally, if a laboratory is reporting like a probe positive, in the past, that was primarily related to the use of AccuProbes, which were used for culture identification. Those are going away. So hopefully, we're going to get rid of a lot of this DNA probe positive terminology. But I can't guarantee that that's going to be across the board. But it's unfortunate with some of the terminology that we use in the lab.
Yeah, I think that's, I guess, just in medicine, sometimes, I guess, in general, it makes things confusing. That's why these lab talks are so important. And I know I do, and a lot of the nurses eat it up. So we're always having these different webinars over and over again, because we just need to constantly be reminded of some of the terms and what we're looking at and the timeframes. The timeframes are extremely important for nurse case management out there, because if you're going through a chart and you're not getting your results back, or you have to have an idea when they're coming back or when you should start looking for them,
Yes,
for sensitivities and things like that. I do have one more question though about your service. We use it a lot here, I believe, in New Jersey for the MDR, and we're thrilled to have that available to us. So now with the fluoroquinolone testing, so you will take specimens for fluoroquinolone testing just because of the INH shortage, is that correct? Like if a doctor's not going to use INH, but they don't feel comfortable putting them on without doing a fluoroquinolone testing, those specimens can be sent to you guys?
So there's two things. We initiated that service both to help with the isoniazid shortage, because fluoroquinolones were going to be added in most of those cases. And then the second one was also to be supportive of programmatic rollout of the 4-month shortened treatment regimen. for drug susceptible pulmonary TB, which includes MOXIE. So it would be both of those situations that you could request. But I'll let Anaska say a little bit more if you want to about what you've received. I think it's been primarily related to isoniazid.
I think, yes, it has been. And if I'm not wrong, I think we might have received one from the state of New Jersey during this time. But yes, it's primarily to address at this point the INH shortage. And there are a couple of specific states that are using our services regularly because of the situation they're in. But yeah, we would accept, and again, as a reminder, we would not, with this type of service, provide a phenotypic DST as well. That's why it's rapid. But if we find indication that there might be rifampin or fluoroquinolone resistance, we are going to reflex. And our preference is for isolates in this case, just because we know that they're gonna really provide the results that's necessary much faster. But that doesn't mean that if there is the need, we're not gonna, accept a sediment.
Okay. And I guess if there's situations outside of the norm, if you put that in the explanation, I guess those are the ones that we could discuss if you would accept, because I know sometimes we have patients that are not MDR, but we have to treat them like MDR because of intolerance.
And yes, and things like that. And for, I should also mention that does not take away the MDR service that we regularly offer. So if you are in doubt or if you don't know what exactly is the best service to, based on your needs, you can contact us and send us an email. We look at this during the whole day and we reach back to submitters and navigate, based on what their need is. And we may start with, the fluoroquinolone rapid testing, or we may say, in your case, it's really indicated that you get MDDR directly. We've had a couple, maybe a little bit more samples that came to us, and they were mixed cultures actually. So the rapid testing really did not give them the answers that they were looking for. And we reflex this to MDDR because it's really indicated MDDR when you have mixed samples.
Thank you.
To help them with the answers.
I know a lot of our docs reach out to you guys, and you're always helpful and help them get what they need. I do have one last question. I know we're at our time, but I'm just going to ask this last question, and then just remind everybody before you go, we are having treatment of TB disease next week, same time. Are there any instances where non-respiratory specimens could be tested for molecular drug-resistant testing?
So for us, our acceptance criteria are as long as the sample is nucleic acid amplification test positive.
And it's a sediment.
And it's a sediment. It's a processed sediment. Typically, if you want to say a little bit, if it's why we have that nucleic acid amplification requirement.
So yes, one thing is that we do not offer TB diagnostic testing here. So we need that part to be taken care of by the submitter. We need to know that this is really MTBC positive, or if it's an isolate, we need to know that this is, confirmed MTBC, whether it's pure isolate or mixed culture. That's one thing. The other thing is that why do we need this to be a sediment? Why cannot we just receive, raw samples? This is because we don't use chemical DNA extraction method. We do heat kill inactivation. So oftentimes when you are taking a sterile sample, but it's not regular, processed and sedimented, it may pretty much coagulate during the heat kill inactivation, and then we can do nothing with it. We need this to be in pipettable form, so that we can, once we heat kill inactivate it, we can add it to our PCR reactions, and that's the first step of our sequencing process. So for our CLIA certificate, the sample type that is acceptable is a sediment.
So Patty, I guess the answer to your question is it depends, right?
Yes.
We can, yes, we can test those, but they have to meet those criteria.
Okay. So that would maybe necessarily require a phone call or a consultation.
Yep, I think that's probably the safest bet to just reach out.
Great. All right. So we're a little bit past our time, but it was so worth it. Thank you guys so much for this great talk. And I believe we'll have slides available. I'm not sure if they have to be approved since you're from CDC, but
We're going to send them right after this presentation.
Because I definitely want a copy of them, and I'm sure our participants would as well. So everybody have a great Friday, and we will see you next week for the treatment of TB disease.
Thanks again. Thanks again, Angela and
having us.
Adenoska.
Thank you. Bye-bye.
Bye. Bye, everybody. Have a great weekend.